Hi, this is Sterling and welcome to MTHFR Support. Today we will be going over the MTHFR gene and peer-reviewed research. Medical disclaimer. You understand that any information is found within our website or this video is for general, educational, and informational purposes only. You understand that such information is not intended nor otherwise implied to be medical advice. What is MTHFR and how is it regulated? MTHFR is the common name for the gene that provides instruction for making an enzyme called methylene tetrahydrofolate reductase. This enzyme acts as a critical switch in your body's one-carbon metabolism, a complex network of biochemical reactions. Its primary job is to convert the folate you eat in the form of 5,10-methylene tetrahydrofolate into 5-methyl tetrahydrofolate, the active usable form of folate. This reaction is the committed step to creating S-adenosyl-methionine, SAM, the body's main methyl donor. Think of SAM as a tiny worker that adds a chemical tag, a methyl group, to DNA, proteins, and other molecules. This tagging process called methylation is fundamental to turning genes on or off, making neurotransmitters, and detoxifying the body. The MTHFR enzyme has a built-in off switch to prevent it from working overtime. It is allosterically regulated by SAM itself. When there is plenty of SAM, the enzyme binds to SAM molecules, which changes its shape and blocks its active site, putting the brakes on further production. Conversely, when SAM is used up and its byproduct S-adenosyl homocysteine, SAH, accumulates, S-adenosyl homocysteine binds to the enzyme and restores its flexibility and activity. This elegant feedback loop ensures the cell maintains a perfect balance of methylation activity. For the MTHFR enzyme to work correctly, it relies on several nutrient cofactors. One is riboflavin, better known as vitamin B2. This is a critical component of the FAG cofactor that the MTHFR enzyme needs to function. Another one is NADPH. This acts as the electron donor for the reaction. And then we have zinc and magnesium. These are general cofactors for many enzymes in the methylation cycle. MTHFR pregnancy and neural tube defects. The connection between MTHFR folate and pregnancy is one of the most well-established areas of research. Neural tube defects like spina bifida and anencephaly occur when the neural tube, the precursor to the brain and spinal cord, fails to close completely in the first few weeks of embryonic development, often before a woman knows she is pregnant. Having an MTHFR genetic variation does not directly cause neural tube defects, but it can reduce the efficiency of folate metabolism. This is why preconceptional folate supplementation is so crucial. Healthcare officials recommend that all women of childbearing age take 400 micrograms of folate daily. For women with a prior pregnancy affected by an NTD, a higher dose of 4,000 micrograms is advised. These recommendations apply regardless of MTHFR status because supplementation effectively compensates for reduced enzyme efficiency. The NIH's MedGen resource specifically classifies neural tube defects folate sensitive as a condition linked to genes in the folate pathway, including MTHFR, confirming that adequate folate can prevent a significant portion of these defects. The clinical impact of this biological mechanism has been clarified by several large-scale analyses published in 2025, which provide a more precise understanding of the risks. A pivotal meta-analysis published in BMC Pregnancy and Childbirth in 2025 synthesized data from 44 case control studies encompassing 7,384 cases and 51,449 controls. The primary focus was on preterm birth, a closely related adverse outcome. The analysis revealed a significant association between the MTHFR C677T polymorphism and preterm birth across multiple genetic models. Notably, the risk was most pronounced in specific populations, such as those of Asian and Indian descent, while significant correlations were not found in Caucasian populations from countries like Austria. This underscores a critical point. The impact of MTHFR variations can vary significantly based on population-specific genetic and environmental factors. Therapeutic strategies, evidence for low molecular weight heparin and targeted supplementation. If an MTHFR variation contributes to a state of hyperhomocysteinemia and hypercoagulability, the clinical question becomes whether intervening can improve pregnancy outcomes. Research suggests that it can. A 2016 study in the Yonsei Medical Journal followed 50 women with a history of adverse pregnancy outcomes, dividing them into those with conventional thrombophilias, like factor V Leiden, and those with novel thrombophilias, which included MTHFR polymorphisms. The pregnancies were managed with low molecular weight heparin therapy. The results were telling. In the novel thrombophilia group, which included MTHFR, low molecular weight heparin treatment significantly reduced the incidence of fetal growth restriction, intrauterine fetal death, and preterm birth. The researchers concluded that their findings point to the need for novel thrombophilias screening and that targeted therapy can be effective. This positions MTHFR-related risk as a manageable condition. The standard of care typically involves a two-pronged approach. First, supplementation with active forms of folate like 5-MTHF and methylcobalamin. Directly addresses the underlying metabolic inefficiency by providing the end products of the block pathway, thereby lowering homocysteine. Second, in women with a history of recurrent pregnancy loss and a confirmed thrombophilia, a physician may prescribe low molecular weight heparin or low-dose aspirin to directly counteract the heightened clotting tendency. A 2025 master's thesis on precision medicine in this context emphasizes that targeted interventions such as vitamin B complex supplementation, methylated folate supplementation, anticoagulation therapy demonstrate improved outcomes in some high-risk individuals. In summary, the peer-reviewed literature from 2025 and earlier establishes that while an MTHFR variation alone may not be the strongest independent predictor of stillbirth, it contributes to a prothrombotic, pro-oxidant environment through elevated homocysteine. When combined with other factors, or when it contributes to a history of adverse outcomes, it is a clinically significant finding. Importantly, the research supports the use of targeted nutritional supplementation to correct the underlying metabolic defect and, in high-risk cases, anticoagulant therapy to manage the clotting risk, leading to improved pregnancy outcomes. MTHFR, environmental triggers, and autism spectrum disorder, a review of peer-reviewed research. The relationship between methylene tetrahydrofolate reductase, MTHFR gene variations, and autism spectrum disorder has been extensively studied. yet misconceptions persist about whether these genetic variations directly cause autism. The peer-reviewed literature paints a more nuanced picture. MTHFR variants are not deterministic causes of ASD, but rather genetic factors that can increase vulnerability to environmental exposures. This distinction is crucial for understanding the true etiology of autism. MTHFR is a critical enzyme in folate metabolism, responsible for converting homocysteine to methionine, an essential amino acid involved in neurotransmitter synthesis. Two common variants have been studied extensively. C677T, which can reduce enzyme activity by up to 70%, and A1298C, which reduces activity by approximately 40%. The role of MTHFR variant susceptibility to developing autism. A comprehensive 2026 meta-analysis examining 22 studies with nearly 14,000 subjects identified a statistical association between ASD and the MTHFR C677T variant. Though no significant association was found for the A1298C variant, however, the researchers emphasized that substantial heterogeneity remained across studies, suggesting that these genetic variants alone do not tell the complete story. A 2025 study exploring MTHFR activity and ASD severity found a significant negative correlation between MTHFR enzyme function and symptom severity. Patients with lower MTHFR activity exhibited more severe ASD symptoms. Importantly, this indicates that reduced enzyme function may exacerbate outcomes when other factors are present, rather than directly causing autism. The gene-environment interaction, how chemicals create toxic loads. The critical insight emerging from recent research is that MTHFR variations become clinically relevant primarily when combined with environmental exposures. A systematic review published in 2021 compiled evidence from 72 studies examining xenobiotics, chemical substances foreign to the body, and autism spectrum disorder. The review found that exposure to air pollutants, persistent organic pollutants, heavy metals, pesticides, and certain pharmaceutical drugs interact with genetic variants, including MTHFR, to modulate autism spectrum disorder risk. 9 studies within this review specifically documented interactions between chemicals and MTHFR variants. The chemicals highlighted induced neuropathological mechanisms implicated in autism spectrum disorder, including oxidative stress, hypoxia, dysregulation of signaling pathways, and endocrine disruption. Why MTHFR variants increase chemical susceptibility. Individuals with MTHFR variations have a reduced capacity for methylation, a core biochemical process essential for detoxification. Methylation is responsible for cellular repair, neurotransmitter production, immune system function, and the elimination of toxins. When people with MTHFR variants are exposed to foreign substances, they have greater difficulty eliminating them, leading to toxin accumulation. This accumulation directly impacts glutathione production, the body's main antioxidant and detoxifier. MTHFR variant carriers typically have lower glutathione levels, making them more susceptible to stress and less tolerant of toxic exposures. The resulting oxidative stress and toxin accumulation can impair neurodevelopment during critical windows, particularly in the first 3 years of life. A 2025 case control study examining homocysteine metabolism in autism spectrum disorder found significantly higher homocysteine concentrations and lower vitamin B9 levels in the autism spectrum disorder group compared to controls. Among individuals with autism spectrum disorder, those with the TT genotype of the C677T variant had a 9-fold increased odds of elevated homocysteine concentrations. Elevated homocysteine is neurotoxic and can induce excitotoxicity via NMDA receptors in neurons. This metabolic disruption represents one mechanism through which genetic vulnerability and environmental factors may converge to influence neurodevelopmental outcomes. Ethnic and population differences. The impact of MTHFR variants on ASD susceptibility varies significantly across populations. The 2026 meta-analysis revealed that African Egyptian cohorts with MTHFR variants had higher ASD susceptibility than Asian or European cohorts in most models. These differences may reflect variant environmental exposures, dietary patterns, or additional genetic factors that modify risk. Clinical implications. Understanding that MTHFR variants confer susceptibility rather than causation has important clinical implications. The same 2025 study that found no direct genetic association with autism spectrum disorder risk nonetheless identified significant metabolic differences between autism spectrum disorder and control groups. This suggests that environmental factors, including dietary folate intake, toxic exposures, and other modifiable variables, may be more directly relevant to prevention and intervention than genotype alone. As one research team concluded, exposure to xenobiotics may be harmful during critical windows of neurodevelopment, particularly for individuals with variants in genes involved in xenobiotic metabolization. The peer-reviewed research consistently supports that MTHFR gene variants do not directly cause autism. Rather, these variants reduce the body's capacity to process and eliminate environmental chemicals, adjuvants, and excipients when individuals with MTHFR variants encounter sufficient toxic load during critical neurodevelopmental windows. The resulting metabolic disruption, including elevated homocysteine, reduced methylation capacity, and glutathione depletion, may contribute to the development or severity of autism spectrum disorder symptoms. This gene environment interaction model represents the current scientific understanding of how genetic susceptibility and environmental factors combine in autism spectrum disorder. MTHFR cardiac issues and cancer. The impact of MTHFR on heart health and cancer risk is largely mediated through homocysteine. When MTHFR function is reduced, homocysteine levels can rise. Cardiac issues, elevated homocysteine is a well-recognized independent risk factor for cardiovascular disease. It can damage the lining of arteries, endothelium, and promote blood clots. By optimizing folate and B12 status to lower homocysteine, you can help manage this risk factor. The relationship with cancer is more complex. Proper methylation is essential for maintaining stable DNA. Disruption in one carbon metabolism can lead to DNA hypomethylation, which can activate cancer-promoting genes or errors in DNA synthesis, both of which increase cancer risk. What you can do to prevent these issues. Based on the research, a proactive approach focuses on supporting your methylation cycle rather than viewing an MTHFR variation as a disease. Here are the key steps. Get tested. Work with a healthcare practitioner here at MTHFRsupport.org to get genetic testing for the common MTHFR gene variants, C677T and A1298C. Testing your homocysteine levels is also a practical way to see if your methylation pathway is functioning optimally. Choose the right supplements. Avoid high doses of folic acid, the synthetic form. Instead, opt for supplements containing the active forms. Folate, look for 5-MTHF or folinic acid. B12, choose methylcobalamin, dinosylcobalamin, or hydroxycobalamin. Eat a folate-rich diet. Focus on whole foods naturally high in folate, such as dark leafy greens, spinach, kale, asparagus, avocado, lentils, and broccoli. Support general health. Manage stress. Support your liver's detoxification pathways and address any underlying digestive issues, as a healthy gut is essential for absorbing these crucial nutrients. We hope you enjoyed this presentation, and come visit us at MTHFRsupport.org. This is the beginning of a series of videos we're going to be doing, and now we're going to connect MTHFR with other genes and how they interact with each other. We hope you enjoyed this presentation and have a great, blessed day.